Lecture 7 of our series, we continue with the organelles and eukaryotic organisms, and this time we will talk about the smooth endoplasmic reticulum and about the Golgi apparatus. So the smooth endoplasmic reticulum is this green membranous stack area here, and the Golgi apparatus is this yellow part here. Smooth endoplasmic reticulum is not just found in the soma of the cell, but in case of neurons, for example, that have these very long dendrite and axons, you can also find smooth endoplasmic reticulum in the periphery, in these dendrites or axons, unlike a rough endoplasmic reticulum, which has only been reported in the soma of the cell. The Golgi apparatus is usually located in the soma of the cell, and all of those membranous stacks are usually in very close proximity as they communicate with each other. So let's go over this, and let's start with the smooth endoplasmic reticulum. The smooth endoplasmic reticulum is again a membranous stack, and you have these membranes that have an inner lumen. And this lumen contains a number of different proteins, such particularly enzymes. It contains ions such as calcium ions. It contains carbohydrates. It contains other molecules that at certain times during metabolic processes of those cells are critically important. For example, calcium is an important internal messenger. However, this internal messenger, for example calcium, should only be released at certain times during the metabolic function of the cell, whereas at other times it should be contained within this membranous structure so that it is not reactive and inappropriately activates a chemical reaction that should not take place at this point in the cell's life. And for that reason, for example, the calcium is stored within the lumen of the smooth endoplasmic reticulum and is only released if there's a specific signal that triggers this release of the calcium from the smooth endoplasmic reticulum. At the same time, there are enzymes within the smooth endoplasmic reticulum, and those enzymes, for example, are important in detoxification processes, and particularly liver cells that are important for detoxification of substances that we ingest, for example, alcohol. And these substances need to be cleaved, and they are contained, the molecules that are important for this, a number of those molecules and enzymes are contained within the lumen. And here this is an example of a liver cell that has all these membranous stacks of the smooth reticulum, endoplasmic reticulum. Here you can see the example of a synapse in a neuron, and what you can see here adjacent to the synapse in the postsynaptic element of the synapse is a part of smooth endoplasmic reticulum that in this case classically contains calcium that is released if the synapse is being activated. So smooth endoplasmic reticulum has multiple functions such as the synthesis of carbohydrates, lipids, and hormones, then to keep calcium at bay and release calcium only at times when calcium is needed. And then it plays an important role in the detoxification or modification, neutralization of toxins that we have ingested. And then the ratio of rough and smooth endoplasmic reticulum within cells varies from cell type to cell type. Hepatocytes or liver cells, for example, have a very high ratio of smooth endoplasmic reticulum because they have a lot of those functions in which the smooth endoplasmic reticulum is involved. If we now move on to the Golgi apparatus, similar to the rough and smooth endoplasmic reticulum, it is also a membranous stack, and it usually consists out of 1 to 100 cisternae. It depends on which cell we are looking at. On average, usually most cells have between 4 and 8 of those membranous stacks that I will show you in the next slide. They're often also referred to, you will come across the name of dictyostome in plant cells. This is a similar organelle within plant cells. And the Golgi apparatus collects, modifies, packages, and distributes molecules and others to other cellular compartments. So let's look at the Golgi apparatus in a little bit more detail. So it works very closely with other membranous structures that we've talked about, like the rough endoplasmic reticulum or the smooth endoplasmic reticulum. And what happens is that the molecules that have been modified or synthesized within, for example, here the rough endoplasmic reticulum. Remember when we talked about the proteins that are synthesized here, and that the transmembrane mode proteins that are incorporated into the membrane of this rough endoplasmic reticulum. This is now transported to the Golgi apparatus. And there are two sides to the Golgi apparatus. One that is the forming phase. You also will hear the receiving phase or the sys phase or the entry phase. This is where those freshly synthesized proteins are being transported and received by the Golgi apparatus. Now those proteins are being modified as they are migrating throughout the Golgi apparatus, and they are being packaged properly, they are being modified properly, and they are being tagged so that once they have migrated through the Golgi apparatus, they are now being released here. And this here is either the maturing phase, you'll hear maturing phase, or it's called the trans phase, or it's called the exit phase. And this is now where those vesicles with those properly modified molecules are budding off and are now transported to their target areas. So if we go through this, we have the receiving side, which can also be called the forming phase or the entry phase. And then we have the discharging side, which is also called the maturing phase, the trans phase, or the exit phase. The transport vesicles carry proteins, lipids, carbohydrates, fuse with this receiving side, and the molecules are being modified throughout their journey through the Golgi apparatus, and then they are loaded into vesicles that are secreted from this maturing side of the Golgi apparatus and are now being transported to their target sites. And this is an electron micrograph, and I want you to become familiar with how this looks like in real life. So this would be an electron micrograph. Here FF stands for forming phase, MF stands for maturing phase. So here is where the vesicles would fuse with the Golgi apparatus and deliver all of those molecules, proteins, to this Golgi apparatus. They now migrate throughout those stacks, membranous stacks, throughout those cisternae, and are being modified and packaged. Here at the maturing phase, where you see the arrows, is where they are being released, those vesicles that contain those molecules, and are now being transported throughout the cell to their target areas. And to put all of this in context, I would like to go with you through what we've discussed so far. So we've discussed that we have our nucleus where the messenger RNA is being released that encodes for certain proteins. This messenger RNA is now being transported to the rough endoplasmic reticulum or to ribosomes that are free-floating in the cytoplasm. There the proteins are being translated. And if it is a protein that is within the membrane, these vesicles are now budding off from the rough endoplasmic reticulum containing those proteins. So these membrane proteins within the vesicles are now transported to the membrane of the Golgi apparatus, which you can see here. They now migrate throughout the Golgi apparatus, and they are now being modified. And ultimately, they are released from this Golgi apparatus on this maturing side here. And depending on what kind of molecules are within those vesicles now, these vesicles can go in different directions. What they've shown here is an example for a vesicle that contains a molecule that is needed on the outside of the cell. A typical example for this would be a neurotransmitter. Another example would be antibodies, that if you're infected by a pathogen, that your body forms antibodies, and these antibodies need to be released into the body in order to fight those pathogens. So something like this would be released into the outside via exocytosis. There, of course, are the other molecules that are needed on the inside, so they would not fuse then with the extracellular membrane, but they would deliver their target molecules to other destinations within the cell. There are vesicles that contain digestive enzymes, and these, for example, would be called the lysosomes. And these vesicles would fuse with any organelles within the cell, for example, that is disintegrating, that comes to the end of its lifespan, and lysosomes would be very important for fusing with those disintegrating organelles and then metabolizing those and secreting the metabolic end products and recycling molecules that can be reused for metabolic processes within the cell.